So welcome all. Good afternoon, good morning, good evening, dependent on where you are. It's a pleasure to present to you today's webinar on our TLA technology and targeted complete next generation sequencing of genes and gene fusions in leukemia. My name is Harma Feitsma and I'm the head of oncology business development at Sargentis. And as such, I'm responsible for our applications in oncology. And I'm very happy that I will be joined for today's webinar by Beat Schafer, who is head of oncology laboratories at the University Children's Hospital in Zurich. So we will start with an introduction on Sargentis and our TLA technology, and then discuss the various applications in leukemia. And the point on the use of breakpoint sequences as a marker for MRD testing will be presented by Beat Schafer. We will end the presentation with a Q&A session. And please note that you can ask questions at any time during the webinar by typing them into the question panel at the bottom right of your webinar screen. And please mention with your question to whom of the speakers it is directed. So our company Sargentis was founded in 2012. And we are based on the university and hospital campus in Utrecht, which is a beautiful historic town in the center of the Netherlands. And Sargentis is a spin-off of the Ubrecht Institute for Developmental Sciences and also from Utrecht University. And with our technology, we analyze samples in service, and that includes everything from sample prep to sequencing and analysis when we deliver a report with all the relevant results. And we also sell kits if you want to do the analysis yourselves. And that can be standard kits or they can be for customized applications of choice. We use our TLA technology for various applications, for example, in transgene and cell line validation, in plant genomics. And today we will focus on the applications in leukemia. We have been working in this area for several years already, but it's a pleasure to mention that we have received the European Horizon 2020 grant last year. And with that grant, we are able to develop this application really a step further. Then I would like to discuss our TLA technology a bit in more detail because it's always very helpful to understand in detail what we are capable of, to also understand the possibilities of the technology. TLA stands for Targeted Locus Amplification. It's a targeted low-cost next-generation sequencing technology. It requires only 2 primers, so 2 times 20 base pair of sequence information to analyze a complete locus. It uses the physical proximity and proximity ligation as a basis of selection. It's compatible with all next-generation sequencing technologies. We ourselves mostly use Illumina, but it's also compatible with Ion Torrent and PacBio technologies, for example. The technology is suitable for multiplexing. And the critical advantages are that it's highly flexible because we need to design only 1 primer pair to analyze a locus. It generates complete sequencing information. It's relatively hypothesis neutral as compared to other amplification and capture-based targeting strategies. And it enables haplotyping. And the basics of our technology have been published in Nature Biotechnology in 2014. This paper describes in more detail how the technology works and also several of its key applications. Now, how does this work in detail? If you see here the locus of your interest depicted as a line of DNA sequence, then with this TLA PCR primer pair that we use to target the locus, we generate sequencing coverage across a region very broad over this locus, and that can be as broad as hundreds of KB of sequence. This is because we use proximity ligation as a basis for selection. And if you start from a DNA that is folded within the cell, we use a cross-linking approach to keep those parts of DNA together that are in real proximity. And that is what is depicted here. So the cross-linking is in blue and the DNA is in gray, and the target region is depicted in red. If we zoom in, this is the target region. So you see that parts of the target region which are in close vicinity will be cross-linked together. We then fragment the DNA, but the cross-linking is still intact. And then we religate the fragments that exist to a new template. And this template is then circularized. And so far we haven't done any targeting, but now the targeting comes in place, because then with the primer pair targeted to our region of interest, we are able to amplify these circles, which consist of random fragments, but all those fragments were originally in proximity of the target region. These circles are around 1 to 2 kb in size, so they are quite easy to still amplify. So then the amplification is done on the target, and then a standard next generation sequencing library prep for the technology of choice is done to be able to generate the sequence reads. And although these TLA template circles are only 1 to 2 kb in size, because the whole process is random, so the folding of DNA in the cells is random in every cell. Also, the cross-linking procedure is random in every cell, and the fragmentation and religation are as well random. We generate a complex library of TLA templates that all consist of a random set of fragments of DNA, but all those fragments were originally coming from the same locus. And this is why we can generate this very broad sequencing coverage across a locus. And with this very broad sequencing coverage, we are able to detect small mutations such as point mutations and small indels, but also much larger structural variations, large deletions, large insertions, amplifications, and also very cryptic and complex rearrangements involving many parts of the genome. So in leukemias, one of the key applications is to detect gene fusions in DNA, because gene fusions in leukemias are important oncogenic drivers. And I show an example here where we targeted a key fusion gene, in this case MLL. You see the coverage on the MLL gene on chromosome 11. But you see also the sequencing coverage on its fusion partner. This is shown on the whole genome coverage plot on the bottom left. So it's clear that we can detect known fusion partners, which is the case here for MLL-T4. But since we will generate sequencing coverage on any fusion partner present, and it's known that MLL can have various fusion partners, we will always be able to detect that fusion partner, also if it's previously unknown. And where other sequencing technologies for the detection of gene fusions just have to rely on sequence reads spanning the breakpoint of those fusions, as you can see, we generate much broader sequencing coverage across the region of the fusion partner, and therefore we can call gene fusions with much higher reliability than other technologies. We can do this on viably frozen cells, which are usually available from leukemia samples. But if they are not available, we can also do this analysis on isolated genomic DNA. And we also have good experience with whole genome amplified isolated genomic DNA if only small quantities are available. As mentioned earlier, TLA technology is suitable for multiplexing. On this slide, I show an example of a multiplex gene fusion detection panel. And the level of multiplexing is currently quite similar to what can be done with normal PCR. So that is several tens of loci that can be multiplexed in one go. But with new amplification multiplex technologies and as well with capture strategies, in the near future, we will be able to improve the level of multiplexing significantly. In this picture, we show a RUNX ETV6 gene fusion, which could clearly be detected from the sequence information. And this was a quite a common fusion, but also more cryptic gene fusions where at the breakpoint insertions or deletions have happened will be easily detectable by our technology. So we used this multiplexing strategy in a project together with the Princess Maxima Center in Utrecht to target the complete set of genes of interest that are relevant for patient stratification and for potential treatment with tyrosine kinase inhibitors. So we created a panel that could detect the classical set of gene fusions, but also the BCR-ABL-like gene set, including several other gene fusions that can happen there, and also mutations in several genes. And then, so this explains that the number of genes that can be targeted is clearly higher than what can be assessed with the standard FISH approach. So for this type of conditions, it's really relevant to have other technologies available. And we use this panel to assess a set of B-cell ALL samples, and we could confirm known gene fusions in this set. But we also detected unknown fusion partners and some new gene fusions. And we could also detect several deletions and single nucleotide mutations in several genes. So that means that all types of genetic variation, all types of mutations can be detected by our TLA technology. For example, also resistance mutations that can happen, for example, in the ABL gene. So since we generate complete sequencing information across the locus or across the fusion gene in this case, we are also able to use our technology to detect the exact breakpoint sequences at the DNA base pair level. And we can use this for several applications. One example I show here is that we detected the exact genomic breakpoint of a TCF3 PBX1 fusion. So for fusion genes, this is obvious. But we can, for example, also detect breakpoints of larger deletions or other structural variations. On the bottom of this slide, I show an example of an Icaros deletion breakpoint, which is also very relevant in leukemias. And these breakpoints are, of course, very individualized markers, so they are very patient and cancer specific, and therefore they can be used for very specific applications. And one is the case that I want to share here. This is a case from the Princess Maxima Center, which we analyzed for Roland Kuiper and his team. It was an ALL patient who was cured successfully, but then after several years, it was again diagnosed with leukemia. And now the question was whether this was a relapse or whether it was a newly occurring leukemia. And this question, the answer to this question was, of course, important for treatment choice, whether treatment could start with the standard initial treatment or whether a relapse treatment was needed, such as bone marrow transplantation. So we analyzed these samples, both of the first and the second leukemia. And what we found is that the breakpoints of the BCR-ABL fusions, because both leukemias were BCR-ABL driven, the breakpoints of these fusions were identical in the first and the second case. So that meant that this was a relapse indeed and not a second leukemia occurring. Another important application for which the breakpoint sequence of gene fusions can be used is as a sensitive and personalized marker for minimal residual disease testing. And this is the part that will be presented by Beat Schafer, with whom we have been collaborating for a long time on this application already. So I will now give the word to Beat.
Thank you very much, Harma. So welcome also from my side. And I would like to start with giving you a small introduction into MRD diagnostics and why we want to determine minimal residual disease in leukemias. So if you can, next slide, please. So why is a measurement of minimal residual disease important? You can see here, I think its value leads mainly because it integrates several different factors. So it integrates treatment response during different therapies and also immunological effects, but it also integrates the cell-specific events like different mutations and just measures outcome in an unbiased way. So in the next slide, you can see its value in clinical studies. So these are data published several years ago. And you can see if we measure MRD levels at different time points, usually we do at 1 month after treatment and 3 months after treatment. And you can see the difference in outcome in the different patients. So if patients have non-detectable disease already after 1 month of disease, we have a very good outcome, that's the green line. And if patients are still MRD positive after 3 months of treatment, that's the red line, then the outcome becomes very bad. So in the next slide, you see various techniques that we can use to measure MRD. First of all, you can, of course, use morphology. Then you can look at the chromosomes, so by conventional karyotyping. And you can do FISH to detect specific chromosomal translocations. You can also use FOX, and especially nowadays with high multiplex FOX, the sensitivity of FOX also is steadily increasing and also becomes more reliable than it was a couple of years ago. But what I would like to discuss today is the quantitative PCR approach, which still is the most sensitive method that is available today. So how are we doing this? In the next slide, you see the principles. We need a specific PCR marker to detect DNA from leukemic cells. And normally we take advantage of the biology of leukemic cells, could be B or T cells. Here in this example, you see the immunoglobulin locus. And during normal B cell development, there is VDJ recombination occurring to generate antibody diversity. But there is not only VDJ rearrangements, but also in the bottom of the slides, you can see that the rearrangement occurs via non-homologous end joining. And so the joining of the fragments is not precise. That means there is random insertion or deletion of nucleotides at these breakpoint sequences. Now, if we assume that leukemia occurs from a clonal origin or from one cell, then in theory, each cell in the leukemia should have the same breakpoint sequences. Whereas in the normal population, all these breakpoint sequences are different. So if you amplify over the breakpoint regions, you get either a smear or a specific band representing just one sequence. Then next, we can use this sequence and sequence information and generate a specific PCR product, which is both leukemia and patient-specific. With this product, we can do quantitative PCR and establish a standard curve. With the standard curve, we have to establish two very important parameters. That is both the quantitative range of the standard curve and the assay, as well as the sensitivity of the assay, which distinguishes specific amplification from the background. And guidelines how to interpret these results have been published by the EuroMRD group. The next slide. You see then such standard curves that we determine, and then you can use the linear regression to monitor and assess levels of leukemic burden at the follow-up samples. Now, of course, this is very important. It will also be carried out in large clinical trials. So in the next slide, you see that we established all these parameters that are needed, and we standardized this in the context of the EuroMRD group. The group contains now, or consists now of 58 laboratories, and we organize 2 quality assessment runs per year. And usually levels of leukemic burden that we detect within all the different laboratories are in the range of half a log. We do this for ALL, but we also do some lymphomas. So now where does TLA come in? The next slide. You can see that this approach works very well, is clinically validated. However, in our experience, this is around 5 to 10% of the samples. We can either not detect a specific breakpoint sequence, or the breakpoint sequence that we detect does not have enough sensitivity. So in these cases, we would be very happy if we could use another breakpoint sequence as a marker. So for these leukemic samples, we then usually carry out normal karyotyping or array CGH, which would indicate us a candidate translocation. So with these candidate translocations, as enable fusion, MLL fusions, and so on, with the information about the candidate translocations, we can send the samples, or we did send the samples to Sargentis for TLA analysis. And usually within about 2 weeks, we receive the sequence analysis around the breakpoints and the partners that are involved in the chromosomal translocations or deletions that we detected by array CGH. And then we are at the same level than we were before. That means we can generate, or we can design a new candidate with PCR assay and then assess the levels of MRD, similar to what we would do with the immunoglobulin locus. In our experience, we so far sent 18 different samples to Sargentis for the analysis, and we had only 1 that did not work. So in 17 cases, we could get additional information that was not possible to get from the normal classical analysis. The next slide. You see how these breakpoints do perform, and you see here a comparison between classical immunoglobulin-based MRD markers and TLA-based breakpoint markers. And for these different breakpoints that are indicated in the bottom, and the 3 patients I'm showing here, you see that the levels of MRD that you measure are very identical. So at least one of the classical markers always shows up with the same level than the TLA-based breakpoint sequence. There is, however, only one caveat here, and that's shown in the next slide. All these breakpoint sequences that you use are, unlike the immunoglobulin-based sequences, are not clinically validated. We would have to do this for every single breakpoint, which is of course not possible. And it's known that some of these breakpoint sequences might occur also in non-leukemic cells. So this has been published for the B-cell able genomic breakpoint, which sometimes gives results that are much higher than the conventional analysis. So as it says here, over 20% of children with B-cell able variants had significantly more than 1 log higher levels of fusion than when measured with the classical immunoglobulin rearrangements. But apart from this, so this is the only example that we have literature on at the moment, and apart from this, in our experience, breakpoint analysis works as well as immunoglobulin analysis. With that, I hand over back to Karma.
Yeah, so thanks very much, Beat, for this clear application. And I want to mention that we're currently validating this approach of using genomic breakpoint sequences further in a retrospective study with the Princess Maxima Center to see how these markers do in relapse situations. So we'll keep you updated on that. I quickly want to touch upon a product that is also relevant for leukemia, which we also work on, and that is on CAR T cell validation. So as you probably all know, CAR T cell therapy is becoming very relevant for leukemia. And what we use TLA for is to validate the sequence of the targeted locus in these CAR T cells and identify those cells that have undergone the correct recombination. So this is an example of a publication that we did together with a group that works on this. And we can show how many of the cells show the perfect homologous recombination, which is usually only a fraction. So it's important to assess that. If you're interested specifically in this application, I would like to refer you to an earlier webinar that we have done on this topic on targeted sequencing of transgenes and integration sites. This webinar can be found on our website, or if you're more interested, just contact us and we can refer you to that, and we can discuss further any applications that you would like to do. So in the last section, in the conclusions, I want to do a quick comparison of our TLA technology to more standard genomic analysis that are currently done on oncology samples. So for example, exome sequencing or targeted panel sequencing is used a lot, as well as RNA sequencing to detect expression levels and gene fusions. Those are very valuable technologies that provide a lot of relevant information, but usually they are not complete. So if you look at exome or targeted panel sequencing, you only find the variants that occur in exons. So you will find some small insertions and deletions, but certainly not all. And the same holds for if you use RNA sequencing to detect gene fusions. You will certainly detect a part of the gene fusions, but for example, if the fusions are expressed only to low levels, or if they are not fusions inside the gene, but they are, for example, promoter fusions, then it's difficult to detect that with RNA-seq. And especially fusion breakpoints you cannot detect with these technologies. So whole genome sequencing, of course, promises to be more complete, but this will only be the case at very high coverage, and that, of course, brings significant costs. So the advantage of TLA is that it's still targeted, so you can do this at relatively low cost, but you can still generate complete information. So this clearly shows the added value of TLA as compared to several of the other technologies. So to conclude, TLA enables the targeted complex next generation sequencing of genes, including all types of variation, such as structural variation. TLA detects gene fusions with known and novel fusion partners, and also more cryptic events. Uniquely, TLA is able to find the genomic breakpoints of these fusions, which, for example, can serve as robust and sensitive markers for MRD testing. And as mentioned in the earlier slides, we provide TLA both as service and in kits. And I want to mention here again that we have received this European grant. And so this grant also gives us the opportunity to start up pilot projects for new relevant applications. So if you have some new applications or new projects in mind, please contact us and we're happy to discuss how we could collaborate on this. If you would like to read more about this topic, I refer you to our website. We have 2 application notes on these topics. One on the targeted complete next generation sequencing of genes and gene fusions in leukemia, and a specific one on the use of gene fusion breakpoints for MRD testing. So you can find these application notes on our website. Nevertheless, feel free to contact us to discuss any projects you would like to do. So with that, thanks for your attention so far. We'll now leave some time to collect your questions, if any, and then we get back to you in a few minutes from now to answer these questions.